spread them amongst your colleagues, etc. And then just to introduce our speaker today. So it's a real pleasure to welcome David Moreno Mateos, who's just joined this department as an associate professor in ecology. David got his PhD from Spain. Then he did a variety of postdoctoral fellowships in Berkeley, in Stanford, and CNRS in France, and has been a research fellow at the Barth Center for Climate Change. And the last few years, he was an associate professor at Harvard University, and then decided to come somewhere better, and has joined us here this week. So I'm sure he's keen to chat to you and get to know people in the Oxford community. He's just been here a few weeks as well, so this was a great chance for him to introduce himself and his work to this community. So thank you, and over to you, David.
Thank you, Javinder. So yes, definitely, I came in like 3 weeks ago, and it's been intense 3 weeks. I finally got my second kid admitted into school today after 3 weeks of work. So it's been a great day, finally. Some of you know about that. It's been a long story. So, but it's good. So I'm very happy to be here. I mean, everything's been good so far, except for the school part, which has been very intense. And basically, I came here trying to also, actually, when I applied to this position, I didn't know about the new Leverhulme Centre of Nature Recovery, and was actually when I was here interviewing, when I realized that center existed, it was like kind of, this is amazing. It was an incredible news for me to see there is an actual whole bunch of people interested in trying to understand how nature is recovering, how to restore ecosystems, and which is basically what I do. Basically, I try to understand how ecosystems, let's say how ecosystems respond to the abandonment of an impact, mostly agriculture, but not necessarily. So we'll be looking at also mining and logging occasionally. And an impact starts when people arrive to a place where there was not people, or the amount of people had a very different use of the land, as is the case of the Norse arriving to Greenland. So Greenland has been populated for 4,000 years by different versions of the kind of widely known Inuit community. There was like several arrivals, but then in 1988, this is the depiction of artist in the mid-19th century, how he envisioned the Norse actually the day they arrived, with Eric the Red leading the expedition to southern Greenland, where they created a few settlements and in the southern part, as you can see here. And there were in total, there was 3,000 people in the peak of the community. And they basically were farmers. So Norse basically grew hay for the cattle in the winter, because the winters are very rough, as you can imagine in Greenland. Those days were incredibly worse than they are now. And they basically grew hay in places like this. As you can probably see, this is the landscape of Greenland. And you probably have noticed that is this kind of strange patch that you can see today. This is actually when we were visiting Greenland a few years ago. This is our tents, as you can see here. And this is a hayfield. So even today, we are talking about 600, depends on the farm, 600 to 1,000 years later, you just visually see the legacy of this farmer agriculture. So this is more or less the size of the field. And here are the ruins of the place where the Norse, the family, usually they were like family communities, usually extended family communities. And this is where it used to live. So this is the kind of places that are incredibly interesting for me, because there are places where you go, there's been an impact of, let's say, an intense impact. So you were here like probably 600 years ago, there were like a lot less vegetation, but it was a place where there's been an impact for a certain period, in this case for 450 years. And then that place has been abandoned for whatever historical reason. And then no one else has lived there afterwards, which is incredibly unusual in a place where a population is dramatically increasing. So there's a few places on Earth where this is happening, and Greenland is one of those. And that's why we decided to go to Greenland. So here, what we basically do is trying to understand how these ecosystems have changed through time. So since the abandonment of this impact. And we are looking into that, trying to understand change in a more complex perspective, in a more ecosystem perspective. So in the way that we are looking at parameters that involve a high amount of complexity. I'll go back to this complexity moment in a minute. But also in an environment that is changing. So all these systems are not going to be are never going to be what they used to be. So it's going to be something else. But having an idea of how this drives us to those new situations can be incredibly useful to assess ecosystem recovery and also to inform restoration, which in the end, that's what we want to do. So in the talk today, we're going to be briefly going through 4 main issues. So the first thing, we're going to be talking about global ecosystem recovery patterns that have been happening. I was glad to hear Dalvin there. There is a whole line of people who is going to be talking about ecosystem recovery. And in those, then we will explore measures of ecosystem recovery, in particular, those measures that involve this complexity. I'm going to stop for a moment in these 2 concepts, the concept of recovery and the concept of complexity, because they are perceived very differently from different people. So by recovery, I mean when you have an ecosystem, someone gets there, let's say it creates a wheat field, and then that wheat field becomes no longer interesting after 300 or 3,000 years. Some places in Spain, in Europe, have really long-term histories of agriculture, and then the system kind of recovers. Basically, it doesn't really recover because it doesn't go back to what it was. It basically changes to something. But my interest is trying to see how that system gets to a point where biodiversity loss and ecosystem function loss is no longer happening, basically. So it's a new system of this kind of some kind of dynamic stability. By the way, the system is self-sustaining and is not kind of accumulating a loss of biodiversity and ecosystem function. And the concept of complexity, I actually started using this concept of complexity when I was reading, I don't know, years ago, a paper on complex system theory, and that they defined complexity as the amount of information that an algorithm has that does something. So the more information the algorithm has, the more complex is the algorithm according to the complex system theory. And I said, this is actually very useful for ecology. So I understand complexity as those metrics that are encapsulating a lot of ecological information, basically. And to do that, you can do that in very creative ways. You can do it in pretty much any way. Usually, well, traditionally in ecology, when you are looking at complexity in ecosystems, you usually refer to networks in many cases. And in ecological networks, complexity is basically related to the amount of interactions that are happening in the ecosystem. And hopefully, in my view, I add also what are the functions that are related to those interactions, how those functions change depending on the structure and the nature of those interactions. And this is from a ecological, let's say, a community perspective, but also from a population perspective. In an ecosystem, I understand complexity as the amount of, let's say, genetic information that exists on one population that is making that population to have higher or lower adaptive potential, which is how better or worse this population will respond to change, basically. So those are the 2 levels of complexity what we are looking at, and we'll be going through both of them in a moment. So finally, we are going to walk through 2 of the fieldwork cases that we have, the Greenland that we already saw, and another one in the central Amazon, where there was another kind of people that lived there for many years and left plenty of agricultural lands abandoned. And they range between 400 to 2,000 years. Well, actually go all the way to 6,500 years old, but I'm going to be focusing on this kind of more recent former agricultural areas. And then finally, we look into a couple of potential applications of all these, let's say, theory or all these observations that we are getting in the field, how that can be translated to actual restoration actions, basically. So the first thing is trying to understand how recovery is happening globally. I mean, I'm not going to talk about global recovery patterns because there are probably a few talks coming in soon that are going to be talking about that, but I'm just going to give you a sense of the global recovery patterns that are happening today. This is a map of basically how farmland has been changing over the last 60 years. As you can see, the orange is basically no change. The blue is basically an increase in farmland. The red is a loss in farmland. The purple is just a mixed and clear trend. As you can quickly see, most of the loss of farmland is happening in the northern hemisphere. Most of the gain in farm is happening in the southern hemisphere, which is kind of reasonable. That's what we basically hear in the news with all the deforestation in the tropics happening all the time. But for me, the interesting part is that there's been a 19% abandonment of the farmlands in the last 60 years. And that is a massive amount of land that naturally, without doing much, is already recovering by itself. So if you overlap the forest change map, which is from the same study, it's basically showing us the picture that forests also having like their own unique trends. Again, green is no change, blue is more forest, and red is less forest. Again, you have more forest, a net gain in forest in the northern hemisphere, and a net loss of forest in the tropics mostly. So these two maps, they are not, I mean, they don't completely overlap, but they are telling you two main trends that are related and highly connected, but not exactly the same in terms of the data that they are showing. So it's like we have a loss of agricultural land and a gain in forest cover. So there is actually a huge opportunity to promote this forest recovery and this farm abandonment and use it also, I mean, also through the means of active restoration, not only like just abandoning land, which in many cases is probably the best because we don't really know a lot on how to restore ecosystems. So the idea basically is trying to see how restoration can be improved in all this land, basically in the future, also responding to all these calls on ecosystem restoration that are happening globally. So with this goal, we did our first study trying to assess globally how restoration has been doing. So I'm going to walk through this graph because we're going to have different versions throughout the talk. So this is any metric of an ecosystem, going back to these simple ways to measure change, which is through biodiversity or soil carbon in the soils. This is time. So this is the measure of that metric. So then at some point, this trend starts, then that lasts for some time, this metric goes down or changes. I mean, doesn't matter. And then disturbance ends, and then this thing basically recovers to something that was similar to what you had before. So this recovery debt concept that we coined is basically this area. The pale area is basically the area between what it used to be and the area that what it has been doing the recovery process. And the dark part is basically what we were able to gather in our study, because I mean, the whole area is really hard to get. So you just get pieces of it. So in general, when we compile data from like over 3,000 restored and recovering ecosystems, we found that on average, usually restored ecosystems have half of the animals and plants in general. Let's say half of the density of the animals and plants. They were 30% less diverse and they were 40% less functional, specifically focused on carbon, nitrogen, and phosphorus cycling. So the even more interesting thing for me was that this pattern was incredibly consistent across biomes. So it doesn't matter if it's a forest, a marine ecosystem, or a river. There is always this more or less similar decrease in ecosystem. I mean, in the abundance of organisms in general and the diversity. And in this case, the data weren't incredibly good for what I would have liked to do, which is I would have liked to see how that change happens in time. And we were only able to collect some data on the time. So you can see, I think the average time for this recovery period is 22 years, which, as you can imagine from an ecological perspective, is pretty much like the very, very start of the process. So the next thing we wanted to do is to see, okay, let's try to reconstruct recovery trajectories. So how this is going to actually, this is changing to time. This is what we're finishing right now. This is our paper we are hoping to submit anytime soon. So this is basically the same graph. So we have time and we have a metric of an ecosystem that is recovering. So this is the reference. So let's say the closest it is to the reference, the more recovered it is. And we just gather data for abundance, diversity, species similarity, and then the carbon cycling and nitrogen and phosphorus stocks in soils, only because it's the only data for which you have enough amount in the literature to be able to get kind of some consistent database. As you can see,
are they recovering from agriculture or is it
agriculture and logging? Yes, only. So we eliminated any other impact. So to make it consistent, in the end, I wasn't planning to say it, but in the end, there was no difference in recovery patterns from a statistical perspective between logging and agriculture, which is surprising. Well, there was minor effects, but anyways. So the thing is that, as you can see, Sorry, I forgot to mention all these kind of pale lines in the bottom are the actual chronosequences that were shown in these studies. We got, I mean, for those of you who don't know what is a chronosequence, is basically a series of plots in an ecosystem that have been abandoned at different times in history. So for instance, you have a plot that was abandoned 50 years, another one 100 years, another one 150 years. And then imagine that you have a reference that this is still being, let's say, impacted. So with that, you build this reference, this chronosequence, and then you compare that against a reference, which is commonly a forest that has been, let's say, ideally not disturbed, but that doesn't really exist, but let's say with a lot less disturbance, or at least for the last few hundreds of years. So this would be an undisturbed, unquote, ecosystem. So the question is, every metric recovers in very different ways. Basically, the main trends are basically statistical models of like a conglomerated chronosequences. And you can see some metrics recover kind of, let's say, quickly. We are talking about 150 years for these metrics. We're talking about 300 years for the abundance of some species. But other metrics like the species similarity is taking a lot longer. So species similarity is a tricky metric, because you are hoping to have the same species that you had before, which is very hard to happen. But as you can see, it kind of tends to happen in some way. So it gives us some information on ecosystem size. So the predictions, basically, I mean, if you cut the slice on the 50 years to give you a sense of how recovery operates, you can see that pretty much every single point that we had in our database was well below the 100%. So this is basically telling you that 50 years is just at the very beginning of the recovery process. So the question now is, what is going to happen in the future with these metrics of recovery? And what is going to happen also when the environment is changing so dramatically, so we don't really know where we are going. And that's the next step of the research that we want to do, is we want to know, we're going to develop some tools to try to predict these recovery trajectories based on current changes in different parameters, that environmental parameters. So the thing is also, I mean, this time scale is not, I mean, I try to make it accurately. So what we are finding when we try to estimate this, we are looking at periods of time which are really long. So for instance, we are finding that to recover the abundance in the forests, we are talking about, in our parameters, time scales of 400 years at the least. So the thing is, it gives you a sense of the time scale of recovery. But we're going to go well beyond that a little later in the presentation. And in reality, you think that recovery takes a long time, but I mean, it's not surprising, because I mean, think about a forest like this one. This is the highlands of Costa Rica. These are oaks, most of them. And this tree has like something between 400 and 500 years. And you can see the huge complexity of the system just by looking at it. And then one kind of logging company comes here, and then it logs it, and overnight, literally overnight, you can have a place like this. So thinking that you can go back easily to that state, I mean, it's clearly not possible, basically, in a very long time. So the question is, as we increase this degradation, let's say, this loss of complexity, forests are actually changing. So this study were showing a couple of years ago how global forests are changing. So this is basically a depiction of a chronosequence. So you have an old growth that for whatever reason is recovering. But the thing is that during this recovery process, there are many factors like putting stress on the system. And they were looking specifically at changes in temperature, droughts, land use change, wildfire, and insect outbreaks. The thing is that through time, these forests, the new forests, are just different forests. And they found specifically that these new forests were younger in general and shorter, literally. So that's the interesting point. The thing is that overall, we are getting younger forests, globally speaking. But the thing is that to understand the deeper, because they were looking at, let's say, like broad scale, high profile ecosystem, I mean, metrics, but looking at the deeper kind of a structure of the ecosystem is what really is interesting for me, because that's what is going to be hard to recover. This graph that probably some of you have seen is basically the first idea of how the complexity of a forest is built. I mean, it's actually very simple, if even it looks complicated. So this is a 50 by 50 plot in Washington State, and these are Douglas firs, and the black dots are sampling points, and each of these weird shapes are fungal genets. For those of you who doesn't know what a fungal genet is, basically, let's say individuals that spread throughout the floor of the forest. Each of these links is the connection between one tree and one of these fungal genets. And there are trees like this one that they call the mother trees were connected through this mycelial network to 32 other trees within the forest. So the thing is that we are looking at one species of trees and just 2 species of mycorrhizal fungi from the genus Rhizopogon. And the crazy thing is that this is the incredibly simplified version of a forest. You should think that you would have, like, I don't know, an average of 3 to 8 species of trees, probably a few dozens of understory species and grasses, and then hundreds. So on average, for temperate forests have something between 80 to 100 ectomycorrhizal fungi species only, and then you need to add all the other fungal species. So I think the picture of complexity is just crazy. So if you go to a place like this and you log it, that you convert it in agriculture, how long is it going to take to recover some complexity like that? And that complexity gives the system specific features that makes that ecosystem and not a very simplified version of it. So under this kind of paradigm, we have a couple of questions. The first, which is the trickiest one, is how do you measure change? I was interested in hearing that some speaker is coming to talk about the holy grail in biodiversity metrics, which I'm so interested in seeing because I don't know how to do it. I'm just exploring things that I think are complex, but in reality they're incredibly simple. So we are basically trying to look at the structure of the interactions, like this one here, and try to bring it a little kind of bigger. So more species to have a sense more on how things interact and how those interactions, as I mentioned, produce the functions of the ecosystem. And then from this population level that I mentioned before, we also want to know how once the impact ends and how the population respond to that end. So as you will see in a moment, as you put pressures on ecosystems, there is a selective pressure on the genomes of those populations that are also eroding, in most of the cases, the genetic diversity, and that genetic diversity will have an effect, mostly decreasing the potential, the adaptive potential of those populations. And that is also the other question that we're going to see, is how those genomes recover as the pressure is released. The second question is, how long does it take this whole process, which is again a question which we have literally no idea, and well, we are starting to have some sense of the magnitude of the issue. So to respond to these questions, we have a few hypotheses that we are trying to test. The first one is basically that the response of these more complex metrics, the response of biodiversity metrics to the recovery process, and also the effect under environmental and also the process of environmental change, which is going to be having the most effect in ecosystem functions. And that's something that we're going to test using a couple of sites. This is the example we have for our Greenland project. And testing this is complicated because you need an ecological layer of information, but you also need an historical layer of information. And the tricky one is, as ecologists, we are very bad at reconstructing the history of places because we don't know how to do it. So that's why I merged disciplines, as I mentioned in this plot. So this is let's say I would be like me talking about how ecosystems recover. This is the same graph we had before. This is time. This is a metric of recovery. This is the reference state. And metrics, as we saw, respond in different ways. Some metrics recover quickly, like spatial richness, and others may recover a lot more slowly, like the structure of the interactions. And this is the chronosequence. So you have, well, actually, this is the chronosequence. This will be the reference, the historic ecosystem. This is the, let's say, degraded ecosystem, and this is the recovering ecosystem. But the thing is, to build a land history here is where you need to team up with others, people mostly in social science. And so, for instance, in the Greenland project, we are teaming up with archaeologists because they can tell us what happened in the land, because we just don't know. So based on their archaeological methods, mostly using carbon-14 and other issues and other methods, we can understand what happened. So, and what happened means when these people arrived, for how long they were, what did they do in the land, and when they died. When Greenland actually they died, in other places they just left. And reconstructing that land history is very difficult. We are trying to do this. We've been trying to do this also in New England, in another sequence of forest recovery that has been happening there, but I'm not going to get into that. And that's why we have been teaming up with environmental historians, because again, it's so difficult. The thing is that since this is happening under environmentally changing conditions, how do you control for that? Tricky part for which I have not a clear response. What we are trying now is also teaming up with paleolimnologists to reconstruct the response of the ecosystems to past climate changes, and see if with that we can have a sense of how we can expect the system to respond in the future. And then the last part, as I mentioned before, is try to see what's going to happen in the future. And here's where we're going to be using, this is not happening yet, will happen soon, looking at what we call the recovery probabilities, which is basically what are the chances that one metric follows a certain trajectory under certain environmental conditions in the future. And this is what I team up with my theoretical ecologist colleagues to predict. So this is basically the picture that we are trying to address, to kind of use to test our first hypothesis. And in these systems, what we are doing more specifically, so where's the fieldwork here? So the fieldwork is basically going to the recovery ecosystem, sampling the plant community and the soil microbial community. And this is my, let's say, simple approach to complexity, to understand the complexity, which, because ideally you need to sample like many more things, but for now we're starting, let's say, with that. So specifically, we're looking at a whole array of organisms that are interacting with the tree, which are like mycorrhizal fungi that have mutualistic interactions, but also the opposite, pathogenic fungi, but also bacteria that interact with the fungi in different ways, and then other organisms that are also feeding on these bacteria and fungi. And this guy gives us a little bit of a sense of how the system underground is actually affecting the system above ground. So with this idea, there was this study that I want to share with you a few years ago that was trying to understand how complexity is related to function. These guys from the Netherlands were looking at these 3 meadows that were abandoned 1 year, 15 years, and 30 years before the study started. They basically did high throughput sequencing, basically what is in the soil, like everything. And all these are groups of organisms in the soil. For instance, all these are bacteria, the red ones, and these blue ones are fungi. And so these are co-occurrence networks. We can have a whole discussion about that, but I'm just gonna give you the idea of what these people found. And they was basically seeing how many links and how strong were they links through time. And they found that through time, as you can see, there were more links, but it was more interesting because in the later stages, there were more strong links. And that was giving these networks a lower vulnerability to secondary extinctions, which basically they were more resilient to change. But not also that, they also found that these networks were more efficient in using carbon, because there was a transition from a bacterial dominance to a fungal dominance, and fungi are more efficient in using carbon than are bacteria. So for me, this is the first very clear, and it hasn't been repeated in that sense, I think, in other places on a larger scale, which is one of the things I'm trying to do, in that how more complex systems are more functional, or let's say they are more efficient in using resources. Sorry. So the next question is this something that happened in this meadow, or is this something that maybe happened in other places? And this basically drives us to our second hypothesis, which is basically trying to see if there are species, what we call MetaCommunity Hubs, I go back to this in a minute, that are showing similar traits throughout kind of, let's say, a latitudinal gradient. So there are global patterns of ecosystem recovery. So if those traits are consistent, or at least kinds of traits are consistent, we can actually use that for restoration. That's kind of the goal of this idea. And meta-community hubs are basically what we call those species that have unusually important roles in the recovery process. And that's exactly the kinds of species that we're going to find for restoration. So these are basically the 3 projects that we have ongoing in the field. Hopefully at one day will be comparable. For now, we are just working independently in each of them, trying to get kind of consistent data. And the issue with this approach is that since the environment is changing very quickly, especially temperature is changing, mostly rising, and these drivers are having very different kinds of effects in this recovery process. So what we are kind of hypothesizing is that, for instance, temperature will have like 2 opposing effects. On one side, temperature accelerates processes, specifically biogeochemical process. But in the opposite way, temperature also increases metabolic rates. And by increasing metabolic rates, in most of the cases, high metabolic rates include more pathogenic interactions and less in mutualistic interactions. So in reality, it's having one positive effect and another potentially negative effect that we don't know what's going to be the outcome. This is basically a hypothesis that we would like to test at some point. And this is from the community perspective. And the next step, as I mentioned before, is what's going to happen with the genomes of the populations that are part of those ecosystems. And this hypothesis is based on basically an assumption that was supported a few years ago from this graph. So this graph was a paper that was showing the global map of genetic diversity. So what these people found is that in this gradient of disturbance, so this is more disturbance, less disturbance, there was an increase in genetic diversity, except for the dense settlements, which is basically like kind of urban areas in most of the cases, which are highly manipulated by people and increase, let's say, is an artifact of human manipulation. When you have when you don't have that effect, then there is a real increase in genetic diversity. So my hypothesis is what happens if we stop these pressures, these disturbances. My hypothesis is that it would actually recover all the genetic diversity. And genetic diversity in itself may not tell us a lot, but genetic diversity is what is actually allowing the species to have more chances to cope with change, basically, which is what we call the adaptive potential, all evolutionary potential. So we're assuming that as genomes recover, the species have more chances to adapt to change, basically. So with these 3 hypotheses in mind, we're going to go to the field and see how we're trying to test them, specifically hypothesis 1 and 3. The one is this the interactions hypothesis, the complexity hypothesis, and then the adaptive potential hypothesis. So the first is Greenland. This is we visited Greenland a couple of times, and we just go to these Norse farms, and we sample the plants, the roots of the plants, and see who is there. Basically, try to build these networks. If you go to classic environmental and classic monitoring parameters like biodiversity, you quickly see that the amount of the local pool of species is pretty much the same. Actually, the species composition is actually not very different. But if you look at but you can see, visually, again, it's amazing. It's really astonishing to see how this field was abandoned, 700 years ago, and it's still incredibly different from the surrounding environment. And this is mostly due to agricultural legacies. So in degraded, let's say in post-agricultural areas, you will have a lot more nitrogen and phosphorus than in the places that were not farmed. And we're talking about like 5 times more nitrogen and almost 3 times more phosphorus. So and this has an impact, a legacy, basically, a legacy impact in these populations as we saw, but it also has a functional effect. So in these roots, what we basically did, we sequenced the roots to see what is the DNA that was present from the fungal communities. In this case, this is basically like a preliminary, these are preliminary results. So what we found is that in places that were disturbed by the Norse, there was a switch from more mutualistic interactions to more pathogenic interactions, as you can see here and here. So the human use of land has basically increased the pathogen interactions and the pathogenic interactions, sorry. And this basically links what the theory and the hypothesis, which the mutualism I have a hard time saying it, sorry. Mutualism meltdown hypothesis that states that disturbed ecosystems have less mutualism and more pathogenism. And the thing is that pattern has been found in many places. So we found the same kind of thing. So the idea is like, okay, if these places, if through time this would go back to the same amount of mutualism, certainly not probably the same species or similar amount of pathogens. So when we looked a little deeper into the interaction, the structure of interactions themselves, we came up with these networks. So this is the plant community. So the larger the size of the block is, the more abundant the species in each of the blocks that we sampled. And this is the fungal community. Again, only fungal community, no other microbes for now. And each of these are basically, let's say, the operational, the species of the fungi. As you can see, in disturbed ecosystems, we have like literally half of the species of fungal species. But we saw that the plant community was exactly the same. So this effect, you would only see it if you dig deeper into the structure of the ecosystem. And these networks, because they were having a lot less fungal species, they were having a lot less interactions, basically what you mentioned here, links. So basically, in the end, these networks end up being a lot more vulnerable to further extinctions. So their ability to respond to change decreases. So the next question that we're gonna do in this project and other projects is trying to see what are the species that are having these key ecological roles, key functional roles, the species that we call the MetaCommunity Hubs. But for that, we need to do many more analysis. So still, we don't know which are those species, because those are the species that we could understand, we could use in ecosystem restoration in the Arctic or elsewhere. I mean, not because the same species in the Arctic you could use elsewhere. The traits that hopefully will be consistent could be found in other species elsewhere, which is basically where I was trying to show with this graph here. So now we are going to go to the second place where we are trying to test our hypothesis, which is in the central Amazon, not far from Manaus. So in the Amazon, you probably know that the land use history has been incredibly intense over the last many hundreds of years. And so over the last probably 2-3 decades, more 3 decades, it's been debated how was there and the intensity of the land use. But honestly, when I was there, we were sampling many places across all the shore of the Amazon and many other tributaries. And in every single place we stopped, we saw the legacy of agricultural uses. And we are talking about primary forests with trees that are 500 years old. So there was a legacy that was coming before those 500 years, and still you can find it in the ground. That was really amazing. And this is what you find. So when you dig a hole anywhere in most of the shorelines, in pretty much most of the Amazon, you find this black upper part, which is what they call terra preta do indio, which is basically a legacy of ancient agriculture, which basically involves more organic matter, very specific kinds of organic molecules that you wouldn't find in the rest of the forest, that are a legacy of agriculture, more phosphorus and more calcium. So, and the thing is that this system is self-sustainable. In some way, it's got to a point where the system perpetuates itself in this terra preta dark mode, so it doesn't go back to the system you had before. So it's incredibly interesting, but I'm not going to get into that because here in these places, what we are now interested is in looking at initially, again, preliminary results, one species, which is the Brazil nut. This is the fresh nut, which is incredibly tasty, way more tasty than the ones you find in the supermarket. And this fruit body is like this big, more or less. It's heavy. It's like 2 kilograms of stuff, and it gets like 20 to 30 different seeds, and they are incredibly nutritious for many reasons. So they've been used by pre-Columbian peoples for at least 11,000 years. So, and it's been artificially, by artificial I mean by humanly dispersed by humans throughout the Amazon for over 11,000 years. So the origin of the species was in one very specific place. So all this human dispersion has been put in and used, it's been putting a selective pressure on these species for larger fruits. I mean, the larger, the better. Actually, if you go to the oldest known populations in the Amazon, the fruits are like tiny little things that nobody really cares about. And if you go to the super selected ones, you will look at these incredibly tasty, huge fruits. And why we got Brazil nuts? Because this is one of the most long-living species that has ever been cropped in the Amazon. So if you want to see the legacy of something of agriculture, I want to see what are the species that will be the last to recover if you actually want to achieve, let's say, full recovery in some way. So in this place, we were sampling Brazil nut trees. As you can see, they are incredibly big, beautiful trees. And we want to see specifically if we can find, well, we were looking at populations that we're hoping to find populations that are showing signs of recovery, of this genomic recovery. So we go to places that there are no history of use at all, and then we go to places where there's been some history of use, and then we try to compare what are the changes. I mean, we don't have a lot of results, the thing yet, but I want to show you what we have, because this is from this week, actually, that we got the first genomic results. So what we do there is we get tissue from the plant, and we do whole genome sequencing, which is basically we sequence the whole genome to see what regions have changed. And that's the key point, because if we identify what regions have changed, then we can actually annotate those regions and know what is being the change. And the change could be the size of the fruit, but they could be also the size of the leaf, what is the stomatal activity, and many other parameters that is going to tell us how the species itself is changing, and if it's changing in a way that is making it more resilient to change, or is got stuck in this selective place that may have happened. So what we have got so far, this is the place where we were sampling. So Manaus is right here, just in case any of you have ever been in Amazon. So this is not far from Manaus. This is, well, it's not far. It's a 4-day boat trip from Manaus, which is crazy. I mean, the logistics in Amazon are crazy. And this is the oldest record of human use of Brazil nut 11,000 years ago, which is an incredibly, what is it? So somewhere around here is the oldest, let's say the center of dispersion. I don't know how to say that in English. The center of dispersion of the species, where, let's say, what the species originated at some point in the history of this species. So from here, that was the center of origin. This is the last year. So it's been dispersed for a very long time all throughout the Amazon here. So we sampled all these 9 locations. So in those locations, we also are trying to reveal the history of the land using, again, carbon-14. And this is actually the time. This is years before present for each of these populations. And the goal here is trying to relate the age of this, let's say the recovery time, the recovery since abandonment, with different genomic parameters. Genetic diversity will be the first. This is what we're doing actually these days. This is a graph from yesterday. So we actually found some clustering in the structure of the populations. Some species, some of the oldest ones have a very different genetic structure than other of the more recent populations. But we still need to make a lot of work. It was just really interesting to see that yesterday. So there start to be some potential patterns, but still this is incredibly preliminary. So the question is now look into the genomes and see what's changing. This is what we are also starting to see. So basically, this is a random chromosome. This is basically the position in the chromosome. The thing is how different populations. I mean, this is basically a metric of genetic diversity, let's say. So how these different populations are having differences in their genome. So for instance, you see this is the former yellow population here is having a really unique genetic structure, but also here you see there is a clear genetic signature that who knows where it's coming from. It could be anything, but this place where this genetic signature of this population is kind of going back to the average, other populations are taking peaks, meaning that there is a region where it's active selection. We don't know exactly what. We'll know at some point, hopefully in a year or so or less. But there is one of these regions that we are interested in seeing how they are changing and in what direction. And then the thing is that you do this for the whole genome. So you get millions of little graphs like this to see what's happening. So then how, my next question is, how this could be useful for restoration, actually, for the practice of restoration? Because I mean, from this, for me, this has a lot of political implications, because the first thing is that if you know that ecosystems take hundreds of years to recover to something that happened before, like, let's say, current impacts, by current, I mean like post-industrial impacts, or let's say 300 years, 400 years. So the regulations need to be also adapted to that time scale. So you cannot think that you can disturb an ecosystem and then you're going, because you can restore it. So that is not happening. That is not going to happen in many lifetimes. And that's one of the key things that I'm interested in saying, and also has implications, because when you see the climate decade, I mean the UN decade on ecosystem restoration and how they are understanding restoration, honestly, it drives me crazy thinking that it's just planting millions of trees, because it's all about people engagement, which is important, and planting millions of trees, which is an incredibly simplistic way to understand restoration. I know that kind of putting all this complexity into politicians today is not easy, but on the other side, there has to be some middle ground. It's that not just planting trees and getting people involved. It has to be something more, something else. Anyways, I'm not going into politics. I'm going to move on to more practical on-the-ground applications. So by doing this whole genome sequencing and understanding what are the regions of the genomes that we are interested in, and hoping that whole genome sequencing is going to get incredibly cheaper in the next probably 10 to 15 years, it's already incredibly cheaper compared to what it was 10 years ago, like literally 10 times less. So it's something that probably anyone can do in the next time, in the near future. So you can just kind of harvest the landscape, looking for where are the right populations to source your materials that have the highest chances to survive ecosystem. I mean, environmental change. So, and I think that if you combine that selection with this population selection with the MetaCommunity Hub approach, which is basically choosing the right species that have key ecological roles in recovery, then you can have, like, let's say, this super species that will basically accelerate the recovery process and have more chances to survive environmental change. The second application is really practical. So the idea here is to create these structures. We call it the restoration pods, which basically you combine all the elements you need to build a micro ecosystem. So once you know where the MetaCommunity Hubs, let's say 3 species of plants, 10 species of fungi, and 5 species of bacteria, and whatever, you actually put all the propagules of these species, hopefully, this is a lot to say, and in different compartments. So you have compartments for these propagules, then compartments for nutrients, compartments for organic matter, and then water or ways to get water in a biopolymer that is environmentally responsive. You just deploy these things in the landscape using kind of this, well, any kind of drone approach. And then those things we are hoping that they can stay in the landscape like kind of dormant for years. And then once you reach the right environmental conditions, which is moisture and temperature, basically, then they release this, they actually mix the components, and then you start this micro ecosystem. It's kind of a wild thing that we are thinking, but actually we are talking with people in environmental engineering and trying to see, and actually people that do this biopolymer thing, which is incredibly interesting, that actually we can actually build this biopolymer thing. The thing is how they're going to respond. Well, there are a lot of challenges here, but basically it's like this complexity approach can be basically encapsulated in this, with this device that hopefully at some point will be kind of could be used in the field. I'm going to close down with just a reminder of the main ideas. Basically, the main idea I always like to tell people is that recovery is a very long-term process, much more than we would think. I mean, if you understand ecology, it's like it's a very long-term process. And to understand how ecosystems change, we need to look at these more complex attributes of the ecosystems. I'm just listing these three because are the ones we've been discussing today. There could be many more. I mean, this is a totally open field for research. So we're talking about interactions. So what is the nature of interactions, the intensity of interactions, and how they are structured, and all these properties are going to make are going to give you a much better idea of how complexity is changing at the community / ecosystem level. And then we can also look into some degree of complexity by looking at these whole genomes of the populations that are important for whatever restoration or conservation reasons. And then finally, how we move from how we convert this knowledge into specific actions that hopefully at some point will be useful is basically using this MetaCommunity Hub approach that is going to basically help you choose the right species for restoration, but also help to choose the populations of those species that are going to be key for restoration. And I think I'm going to leave it here for today. So if you guys have questions, comments.
Thank you. Questions? Anybody have questions?
So maybe we can share the slides because I've been seeing people taking pictures. I don't know if you guys do that or not.
Yes.
Okay. Yes. Hold on. I think we need the mic. The mic. What is it?
Yeah. Who was asking? Sorry.
Thank you. Because you're sequencing, how would you define a population with high adaptive potential compared between these different sites?
Yes, well, that's a great question. I mean, I would respond to that question at two levels. So the first level is, in principle, higher genetic diversity gives more chances for the population to respond to a wider array of environmental conditions. So by itself, having higher genetic diversity could mean higher adaptive potential. But more specifically, I would mostly understand higher adaptive potential if you can identify those regions that are related to the ability of the species to adapt more easily to environmental change. So not necessarily higher diversity, but it's the regions or the functions that are related to their capacity to adapt are in the, let's say, have the right structure, basically. So for instance, think of if you are looking for a species that are more resistant to drought because increasing temperatures are reducing precipitation. So you're going to find higher adaptive potential. In that particular case, it would be a population that have more chances to have, let's say, the ability of closing the stomata more quickly in a reduction of moisture, for instance. And that would be like more specific and more targeted approach to understand adaptive potential.
Thank you.
Question from a social scientist. Is there any evidence that removing human disturbance, for instance, low-level disturbance, mowing of alpine meadows, actually would reduce biodiversity? And what's your thoughts on that?
That's a hard question. I don't really know a lot. I mean, removing an impact can have completely different impacts. In some ways could increase biodiversity, and in some ways could completely decrease biodiversity. And it all depends on the local conditions of the site. And so in the case of alpine meadows, I don't know, because I'm sure the response is going to be very specific to the location where you do that. Because removing the impact of mowing and there is no herbivory or nothing at all, then you are most likely going to have a completely different ecosystem. I suppose if you remove mowing, but you still have local herbivores, there's going to be a very different kind of, let's say, change process. So I wouldn't know, honestly.
Just wondering, yeah.
Thanks. You've done an impressive job at looking at a lot of metrics of recovery, but I wonder if maybe you don't need to go on with that complexity. You could make life easy for yourself by just going back to the old-fashioned metric, which was a community analysis, like an ordination or a twin span. So you're looking at the species composition and the proportions.
Yes.
And that's what we've used as a target in restoration in Britain, just aiming for the original community structure.
Yes. No, I think, I mean, I have this one. So that's actually that's actually a lot better than just looking at number of species or carbon storage and these kind of more traditional approaches. But the thing with species composition is basically that it doesn't give you a sense of how function is changing directly. And I think, and the other thing is that the species that are gonna exist in the new place are most likely gonna be different, not necessarily meaning that those new species are going to be less the typical generalists that come from all over the place and we have the same meadow all over Britain forever, but still it's going to be different. So in some way, this complexity approach, it going to go beyond species composition, because, and this is a discussion I always have, is like, it's not only about looking at interactions to understand functions. I want to understand exactly what species are having what functions. So taxonomy for me, it's incredibly important. This is my fight with many ecosystem functional ecologists, because it's like all about functions, even if they are incredibly complex functions. But I agree that taxonomy is incredibly important. So I always argue that let's look at species composition, but also let's look at what is the functional approach of that and how those species interact in.
Hi, thanks for the talk and welcome to the department.
Thank you.
Yeah, I really loved all of the things that you were talking about with the connection with the above ground and below ground interactions. And I had a question which sort of popped up when you showed the sort of large oak trees and then you showed how quickly they can be degraded. I also work in the Amazon area region, so I see it all the time. But I never really thought about what happens to, like, once you say you don't use fire, and you remove all the trees, that structure and all of the sort of connectors and all of the other species and fungi and things like that that are still below ground in existence, do they still survive? And if they do, if you replanted fairly quickly after a deforestation event, would they be able to reconnect with the young trees?
So what I would say, it depends on the magnitude of the impact. Let's say if you just log the forest and you go and you don't come back ever again in hundreds of years, probably recovery will be incredibly fast, especially if you have a matrix of surrounding forest, because there will be lots of propagules coming in and then it will recover. And even if you don't burn, I mean, usually in the Amazon, every single time they burn, so they do not burn, because basically because that brings like quick nutrients to the soil. But even if you don't burn, let's say, and you convert it into an agricultural field, and that if you break all this structure of the soil just by plowing the soil once, you are breaking the structures that may take a few hundreds of years to recover. So the thing is, it depends a lot on the impact. So it's not the same logging and or burning and or plowing and adding fertilizers. So it's a huge array of disparities. So in the global analysis that we did, that was showing these recovery trajectories, we were kind of finding, still we need to do some more analysis, that forests that were recovering from agriculture were recovering better than forests recovering from logging, which was like, this is crazy. And we ran different kinds of approaches, analytical approaches, and it was surprisingly consistent. And we never will know why this is happening, but you can have your own hypothesis. And we were thinking that as you log a forest and you extract a lot of the resources, the kind of nutrients can decrease. And agriculture may have the opposite effect. When you leave a field, there is loads of nitrogen and phosphorus, and they may be accelerating the recovery process. I mean, that's a hypothesis that we are not going to be able to test. But this is basically to give you an idea that, I mean, the process is incredibly stochastic in most of the cases.
Could this sort of analysis be used like for the advantage of the argument of selective logging, or like, you know, agroforestry or something like that?
Well, I don't know. So how would you use that for selective logging?
Well, I'm wondering, so for example, if the trees that are taken out like in whatever systematic way they choose, which is what they tend to do in like concessions and things like that in the Amazon. technically speaking, you'd still have some of the older trees remaining, and then the underground system would obviously, I'd imagine, reduce,
Be
or
less
it would,
impacted. Yes.
yeah, and it would be less impacted, and
Mhm.
potentially the new trees that start to grow, would they be able to sort of connect with that?
Maybe, definitely. I mean, the lower the impact, the more chances I would say it has
Mm.
to recover, or at least to recover more quickly. But again, I don't know.
Thanks.
Yes. Hold on, I think
Thank you. Very interesting. We had a paper last year where we argued that for a site in Borneo, which is logged, that many aspects of ecological function were amplified, enhanced by the disturbance, and actually then including species abundance, because there were more nutrients flowing through the system, more insects, then more insectivores, and it's cascading through. And so I'm not saying that's general for all logging, but that's what we found on that particular site. But building a little bit perhaps on Florida's comment, there seem to be some types of disturbance that actually amplify, enhance ecological function. So I'm wondering what comes through in your meta-analysis. When you talk about recovery, there's always an implicit assumption there that the optimum state for these metrics is the final end state that you're going to. But for many things, and you see this, I guess, in your carbon and phosphorus cycling, you can actually have amplified ecological features in that disturbed state, and actually the low disturbance state ends up being a regression of some ecological attributes. I just wonder whether,
yes,
given your meta-analysis, whether you had any thoughts or comments on that.
Where we actually, let's say, we nullified the amplified effect, because the amplified effect is also an impact. So that having more species is not necessarily positive, it's just a different ecosystem. So we nullified by saying that having, let's say, 5 more species have the same detrimental effect as having 5 less species, because you don't, and there's no argument to say that that's positive. So the thinking that is positive is basically a mental effort thinking that is positive. It's just different. And so that's why we nullify that effect, because in reality, it's just a different ecosystem. So an ecosystem where there is more nutrients, even if it has more biodiversity or more ecosystem function, in this conceptual idea, it would be still a degraded ecosystem, because it's as degraded as one that has less, but just in another direction. That's how we see that.
Yeah.
That's why I see that basically is that's why I mentioned this recovery concept at the beginning. It's not about recovery, it's about how things change.
But isn't that perhaps a value-laden thing as well, to say that no disturbance is the ecological perfect state, and
It's not perfect, it's just the end of
the ideal state. Whereas actually, if you look at ecological processes, in many cases, then the disturbed system may have more ecological functioning than the non-disturbed system.
It is, but that's not necessarily positive. That's the thing. It's just a different ecosystem. If it's good or not, we don't know. I don't know if it's good or not.
Sounds like a good one for the drinks.
Yes.
Okay.
I think we have maybe one more question, or no, I don't know, whatever you guys say.
Yeah, thanks. Partly following up on Yadvinder's comments, questions, but first, so in the case of Washington State and Douglas fir, my understanding generally in many parts of the state, Douglas fir is not considered a climax species, and we used to talk about that way, because they tend to be occurring in areas where there's a fair amount of natural disturbance. I mean, however you define that, fire, wind, or especially fire, I think, in there. So sort of in extreme fires over a certain cycle. So I would think that how ecosystems respond would vary depending on whether on this sort of natural disturbance pattern. So that's one question. And if you found that, so like, and also that's been a logic with, sort of logging strategies, is that when you need, Douglas fir requires a lot of light, so you need a larger clearance if you want Douglas fir growing back. So I'm just wondering how those kinds of, because they have a higher turnover as natural ecosystems. So that's one question, is do these recovery processes partly depend on your natural disturbance regime? But another is also following up on, I think what Yadvinder was saying there, is like, I think, yes, they're all, I mean, using the word recovery, I mean, does seem to have a sort of normative communication that this is that this is recovering to a desirable state, but you don't mean it that way. You just mean getting back to where it would be if people were not here. If the ideal world is that people aren't here, then recovery means getting back to a place where people are not there. So those are sort of two different questions.
Yes.
Yeah.
Well, the first question, I mean, that wasn't a recovery study. It was just these studies and trying to understand kind of mycelial networks, and just use it as an example of forest complexity without any relation to the recovery process, and basically to give an idea that even those simple ecosystems, with all this, looking at these simple attributes, there is a lot of complexity going on. And definitely these systems are entirely kind of driven by disturbance. So disturbance is part of the thing. They've been living with fire or with other kind of human disturbances for, well, not much, like 15,000 years specifically in Washington state. So that wasn't, I mean, my idea was not to say there has to be no disturbance. And it's just an example, basically, of forest complexity. And the recovery question, that's what I was mentioning at the beginning, my clarification about the recovery. It's not that we want a world where there's no people. That makes no sense. But I always like to have a reference on how a system would be behaving if they would have like no human impact, or at least no heavy human impact. Because that could teach us how, if we need to restore, what we need to be looking at if we're going to restore ecosystems that are as resilient as the natural ones. We may end up finding that all these natural ecosystems and disturbed ones respond worse than, let's say, impacted ecosystems. But in reality, we don't know. So the idea is not going back to an unpopulated place, basically. Although I think the patterns are showing the land abandonment is going to keep increasing over the next decades. So I think it's what's going to happen with all that land, where people is not going to be intensively managing. And agricultural technology is becoming incredibly better by the year. So there's going to be less and less land needed. So how I personally envision the future is like a lot more land abandoned and a lot more land that has much more intense impacts in less surface area, let's say. Well, that's basically, that's a possibility. It doesn't have to be that way, but that's how I see it. That's why I think understanding recovery is going to be incredibly important, as all this process could potentially happen in the future.
Okay, I think we can wrap up there.
Okay.
So, for those of you here, we had about 30 people online as well, so there's a reasonable online audience as well.
Great.
So for those of you here in person, you're welcome to linger and have a drink and an informal chat afterwards. Thanks, Stephen, for setting up the drinks, and I'll see you all next week for EJ Milner Cullen seminar. So thank you again, David.
Thank you, guys.